Hello students. Today let's discuss the formation of urine within the kidneys. In the last class we discussed about the structure of the kidney as well as the structure of nephron in detail. Now let's see how is urine formed within the kidneys. As you know, inside the kidneys there are millions of nephrons. And each nephron has two main parts. The first part called as the Malpighian corpuscle or the renal corpuscle or the renal capsule, and the tubular part known as the renal tubule. Now, the first part, that is a Malpighian capsule, has a C-shaped structure called as the Bowman's capsule, and inside that we find a tuft of blood capillaries called as the glomerulus. Now the glomerulus receives blood through a wide efferent arteriole, and the blood after passing through the glomerulus, it leaves the glomerulus through a narrow efferent arteriole. Now within the kidneys, urine formation takes place in three different steps. The first step is called as glomerular filtration. The second step is called as tubular reabsorption, and the third step, tubular secretion. So these are the three major steps in the process of urine formation. Once again, first step, glomerular filtration. Second step, tubular reabsorption, and the third step, tubular secretion. Now let's see one by one. The first one, glomerular filtration. From the term itself, it's very clear. The filtration of blood takes place within the glomerulus. And you know the glomerulus is found within the Bowman's capsule. Now, the first step in urine formation is the filtration of blood, which is carried out by the glomerulus and is called glomerular filtration. On an average, 1100 to 1200 milliliters of blood is filtered by the kidneys per minute. In a minute, the kidneys filter about 1100 to 1200 ml of blood, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. So when we studied about the circulatory system, we have seen that the heart pumps the blood, isn't it? So there in a minute, that is cardiac output, we have seen that almost 5 liters of blood will be pumped out by the heart per minute, right? So this kidneys filter about 1/5 of that. Kidneys filter about 1100 to 1200 ml of blood, and this filtration which take place in the glomerulus is called as glomerular filtration. Now let's see the definition of glomerular filtration. It is defined as the filtration of blood under high pressure. This pressure is called as glomerular filtration pressure. The word pressure, last word is hidden by that picture. So glomerular filtration pressure, when the blood flows through the glomerulus from a wide efferent arteriole to a narrow efferent arteriole. So now in the picture you can see this is the Bowman's capsule. This yellow colored cup, that is a Bowman's capsule. And inside that we find the glomerulus, which is a glomerulus, that is the tuft of blood capillaries. This is a glomerulus. So blood enters the glomerulus through this wide efferent arteriole, right? So the blood is coming through the wide efferent arteriole. It is passing through the glomerulus. Now inside the glomerulus, that is inside the Bowman's capsule, there is a membrane. A membrane will be formed. 3 layers will be. Filtration takes place through 3 layers. One is the lining of the Bowman's capsule. another one is the basement membrane, and another one is the lining of the glomerulus. So through these three layers, the blood gets filtered, and the inner lining of the Bowman's capsule is lined by certain specialized cells called as podocytes. Podocytes. And these podocytes are arranged in an intricate manner so that they form small pores or slits. So small pores or slits are found in the Bowman's capsule. So the filtration takes place from the capillaries, the blood gets filtered and it comes and gets collected in the lumen of the Bowman's capsule. Now, as you all know, when just imagine, bring to your mind a hose pipe through which you water the plants. So one end of the pipe will be connected to the tap, and the other end, the water will be flowing out through the other end. So when you are watering the plants, if you apply a pressure at the other end, what happens? The water will be pumped out in the form of a fountain, isn't it? So just like that, when there is a difference in the width of the tube through which the fluid flows. Here in this case, when the blood flows through a wide efferent arteriole and it is leaving out through a narrow efferent arteriole. So automatically a pressure will be created in the center portion. That is, pressure will be created in this portion of the glomerulus. So what happens? And if there are small pores in that, it gets filtered. The water gets, the blood gets filtered, or the blood comes out, and it gets collected in this Bowman's capsule. Okay, so that is how the filtration takes place within the glomerulus. So when the blood flows through a wide efferent arteriole and when it leaves out through a narrow efferent arteriole, a pressure will be created, and that pressure is called as the glomerular filtration pressure. Now, as a result of this, water, urea, salts, amino acids and many other substances are filtered into the Bowman's capsule, and this is called as the glomerular filtrate. Means the filtrate which gets collected in the Bowman's capsule, that is called as the glomerular filtrate. Hope you got it right. So glomerular filtrate contains what? It contains essential substances as well as non-essential substances, right? It is not that only the nitrogenous waste will be coming out. Along with the nitrogenous waste, the useful substances like glucose, amino acids, salts, etc. also will be coming out, and it gets collected in the Bowman's capsule. Now, glomerular filtration is also called as ultrafiltration, because all the constituents of the plasma, that is the blood, all the constituents found in the plasma except proteins get filtered. It doesn't mean that the whole blood, content of the plasma will be coming out. When it is flowing, during its flow, some amount of these particles, some amount of these substances will be coming out, gets filtered into the Bowman's capsule, and this is called as glomerular filtration. Hope you all understood now what is glomerular filtration. So this is the first step. So as I've told you, the glomerular filtration, during glomerular filtration, both essential substances as well as non-essential substances will get filtered, and it gets collected in the Bowman's capsule. So automatically the useful substances have to be reabsorbed. Otherwise, what will happen? All the useful substance of the body will also come out of the body in the form of urine. So that should not happen. So the next step is tubular reabsorption. Why is it called as tubular reabsorption? Because this takes place in the tubular part of the nephron. You know which are the parts of the tubular parts of the nephron. The first part is proximal convoluted tubule, or PCT. The second part is the Henle's loop. And the third part is the DCT, or the distal convoluted tubule. So now, the process of absorption of useful substances from the filtrate into the peritubular capillaries is called tubular reabsorption. At this point, you should remember what is peritubular capillaries. So, suppose this is the Bowman's capsule, right, and this is the tubular part of the, this is the U-shaped Henle's loop, right, and then this will be the PCT. Okay, now inside this what is there? Inside this there is the blood. Blood flows out, right, it enters through the efferent arteriole, right, and then it leaves out through the efferent arteriole. The efferent arteriole, after coming out, what happens? It branches and rebranches and form a network of capillaries around the tubular part of the nephron, isn't it? And this network of capillaries around the tubular part of nephron is called as the peritubular capillaries. It's difficult for me to show with this brush. So hope you understood. These are the peritubular capillaries. That is the efferent arteriole. After coming out, it branches and rebranches. What you find in red color is the tubular part of the nephron, and what you find here in green color is the peritubular capillaries. So the blood, after coming out from the efferent arteriole, will be flowing out through the peritubular capillaries. Okay, so blood will be there in the peritubular capillaries. Now, the glomerular filtrate is also coming down. Okay, so the glomerular filtrate, which gets collected in the Bowman's capsule, will be coming down through the tubular part of the nephron. So what I am showing in blue is the glomerular filtrate. So when it is coming through the tubular part of the nephron, what happens is the useful substances which are present in the glomerular filtrate will be reabsorbed into the peritubular capillaries. Useful substances will be moving into the peritubular capillaries, and those harmful substances which have escaped this glomerular filtration and which will be flowing through this efferent arteriole will be through the efferent arteriole will, so the glomerular filtrate is what I am showing in blue color. Okay, right, and so from the glomerular filtrate, useful substances will be absorbed into the peritubular capillaries. So this process is called as tubular reabsorption. The process of absorption of useful substances from the filtrate into the peritubular capillaries is called as tubular reabsorption. Right. Now, for example, substances like glucose, amino acids, sodium ions, etc. in the filtrate are reabsorbed actively, whereas nitrogenous wastes are absorbed by passive transport. So now what is the difference between active transport and passive transport? Active transport is where energy is required. It is not against concentration gradient. Now what is, if there is a difference in concentration of certain substances, automatically from a region of higher concentration, the substances move into a region of lower concentration, isn't it? So for example, here in the peritubular capillaries, the nitrogenous waste will be very less compared to that found in the glomerular filtrate. So the nitrogenous waste will come out without any difficulty. It comes out into the peritubular capillaries. It's not actually required. Whereas useful substances will be found in the glomerular filtrate as well as in the peritubular capillaries. But the useful substances have to be reabsorbed. So it has to be pushed out from the glomerular filtrate into the peritubular capillaries against the concentration gradient, and hence, so what happens, energy is required. So that is known as active transport or active reabsorption. So the useful substances will be reabsorbed actively, whereas nitrogenous substances will be absorbed passively. Reabsorption of water also occurs passively in the initial segments of the nephron. Hope you understood what is tubular reabsorption. Now coming to the third step, that is tubular secretion. Tubular secretion is the process of secretion of non-essential substances. Non-essential substances means substances which are not required by the body, that is the nitrogenous waste materials, certain drugs, hydrogen ions, potassium ions, creatinine, then toxic substances which are not required by the body will be secreted from the peritubular capillaries into the nephric filtrate. That's what I said. When the blood, certain harmful substances may escape, they will not get filtered during the glomerular filtration. They escape and they come out into the peritubular capillaries. So from the peritubular capillaries, those substances have to be secreted back into the glomerular filtrate, and this process is called as tubular secretion. The substances like hydrogen ions, potassium ions, ammonia, creatinine, parahippuric acid, toxic substances and drugs are secreted into the peritubular capillaries. So tubular secretion is just the opposite of tubular reabsorption. In tubular reabsorption, what happens? From the glomerular filtrate, substances will be absorbed into the peritubular capillaries, whereas in tubular secretion, the substances from the peritubular capillaries will be secreted back into the glomerular filtrate. So it is just the opposite. So these are the three main steps in the process of urine formation. So after this, that is after these three major steps, glomerular filtration, tubular reabsorption and tubular secretion, reaches the end of the DCT will enter the long tube which is called as a collecting duct, and the collecting duct of many nephrons join together, converge, and finally it goes out in the form of urine through the ureters, and it gets collected in the urinary bladder. And per minute about 125 ml of glomerular filtrate will be formed. We have seen that per minute about 1100 to 1200 ml of blood gets filtered, and from that 125 ml of filtrate will be formed per minute. That means about 180 liters of glomerular filtrate will be formed in a day. But do you think that 180 liters of urine is coming out? No, it is hardly 1 to 1.5 liters of urine only. So what happens? This 180 liters of glomerular filtrate will be concentrated or will be reabsorbed, and it will be converted into just 1.5 liters. So 180 liters, from that almost 178 liters will be reabsorbed back into the body. Okay, so this is how urine will be formed. So if you get a question, explain the process of urine formation, you have to write about these three major steps: glomerular filtration, tubular reabsorption and tubular secretion. Hope you all understood. Right, now let's see the functions of the different parts of the nephron. Okay, so we have studied about the function of the Bowman's capsule and the glomerulus. Its main function is glomerular filtration. So now here we mainly concentrate on the functions of the tubular part, that is the tubules. The first part, that is the proximal convoluted tubule. It is lined by simple cuboidal brush border epithelium which increases the surface area for reabsorption. Nearly all the essential nutrients, that is glucose, amino acids, electrolytes, etc., and 70 to 80% of the electrolytes and water are reabsorbed by this segment. So it is a PCT which plays a major role in the process of reabsorption of useful substances, that is nutrients, water as well as electrolytes. PCT also helps to maintain the pH and the ionic balance of the body fluids by selective secretion of hydrogen ions, ammonia and potassium ions into the filtrate, and by absorption of bicarbonate from the filtrate. Okay, so that is the function of the PCT. Now here this is a diagram to show the reabsorption by different parts of the nephron. So in this diagram you can clearly see this part is the glomerulus where filtration takes place. Now this part is the PCT, right? This part is the PCT. Now in the PCT, right, in the PCT, here you can see some arrow marks are outside and some arrow marks shows that the substances are going inside, that is tubular reabsorption as well as tubular secretion. So here you can see bicarbonate ion, HCO3 bicarbonate, then NaCl, then water, nutrients, then K positive ions. All these substances, that is bicarbonate, sodium chloride, that is electrolytes, water and nutrients, all these will be reabsorbed from the glomerular filtrate. So glomerular filtrate is the filtrate which gets filtered. So the glomerular filtrate will be moving through this inside the tubule. What you find is a glomerular filtrate. So from the filtrate, these substances, that is HCO3 minus, NaCl, H2O, nutrients, potassium ions, will be reabsorbed back. What is there here? This is an interstitial fluid. This part is an interstitial fluid, right? And whereas hydrogen ions and ammonia will be secreted from the peritubular capillaries into the glomerular filtrate. So this is the function of PCT. Now when it comes down to the Henle's loop has a descending limb. So this is the descending limb of the Henle's loop. Now the descending limb is permeable to water and impermeable to electrolytes. What does it mean? It allows the water to come out into the peritubular capillaries or into the interstitial fluid, whereas it won't allow the electrolytes to come out, that is NaCl or bicarbonate ions. It won't allow sodium ions and other electrolytes to come out, because the descending limb is impermeable to electrolytes and permeable to water. Whereas the ascending limb, this is the ascending limb of the Henle's loop, right? This ascending limb is permeable to electrolytes, right? NaCl, NaCl, whereas it is impermeable to water. So water will not come out from the ascending limb of the Henle's loop. So these two limbs, that is the descending limb and the ascending limb, act antagonistic to each other. They act in opposite ways. That is, a counter current will be created in the Henle's loop. Okay, so the descending limb is permeable to water, whereas the ascending limb is impermeable to water. The descending limb is impermeable to electrolytes, whereas the ascending limb is permeable to electrolytes. What's the meaning of that? Permeable means allows the movement. Okay, now this part is the followed by the DCT. So as a glomerular filtrate passes through the PCT, and when it reaches through the descending limb, what happens? Major part of the water will be reabsorbed, and what remains inside the glomerular filtrate will be concentrated, right? And whereas when so the osmolarity increases, and when it passes through the ascending limb is permeable to electrolytes and impermeable to water. So in this limb, what happens? The electrolyte starts coming out. So again it gets diluted. The filtrate inside gets diluted. Now and as it passes through the DCT, again what happens is reabsorption of water takes place, right? Reabsorption of electrolytes takes place. So again the filtrate gets concentrated. So the urine which comes out, the filtrate, glomerular filtrate, which comes out from the glomerulus, that is from the Bowman's capsule, by the time it passes through this Henle's loop and finally when it reaches a collecting duct, it will be concentrated urine. In addition to this, the peritubular capillaries which is found around the Henle's loop, right? So a network of peritubular capillary found around the Henle's loop, that is called as the vasa recta. So this parallel loop which is found around the Henle's loop, which is called as a vasa recta, also act in a counter current manner, right? So there from the peritubular capillaries, secretion takes place, absorption from the glomerular filtrate and secretion of harmful substances from the peritubular capillaries into the glomerular filtrate takes place in the vasa recta region. Okay, so this two, the action of the Henle's loop and the vasa recta creates a counter current mechanism resulting in the formation of concentrated urine. Hope it's clear? Right. Now, the explanation part of that. So, Henle's loop. This region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The descending limb of the Henle's loop is permeable to water but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. But the ascending limb is permeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrate moves upward, it gets diluted where in the ascending limb due to passage of electrolytes to the medullary fluid. The distal convoluted tubule helps in the conditional absorption of sodium ions and water. It is also capable of reabsorption of HCO3 minus, that is bicarbonate ions, and selective secretion of hydrogen and potassium ions and NH3, that is ammonia, to maintain the pH and sodium potassium balance in the body. So DCT helps in maintaining the pH as well as the sodium potassium balance in the blood. The collecting duct extends from the cortex of the kidney to the inner parts of the medulla. Large amounts of water could be reabsorbed from this region to produce concentrated urine. So this is how the glomerular filtrate gets concentrated and transforms into urine. It also plays a role in the maintenance of pH and ionic balance of the blood by selective secretion of hydrogen ions and potassium ions. So that is all about the functions of the different parts of the tubular part of the nephron. Now the mechanism of concentration of the filtrate. Mammals have the ability to produce concentrated urine. The Henle's loop and vasa recta plays a significant role in this. The flow of filtrate in the two limbs of the Henle's loop is in opposite directions and thus forms a counter current. Hope you understood what is that. That is how when it flows through the descending limb, it is permeable to water. Descending limb allows the water to come out, but it won't allow electrolytes to go out. When it passes through the ascending limb, it allows only the movement of electrolytes and it won't allow the water to come out. So this is what is called as counter current. The flow of blood through the two limbs of vasa recta is also in a counter current pattern. Vasa recta is the parallel network of capillaries, peritubular capillaries found around the Henle's loop. The proximity between the Henle's loop and the vasa recta as well as a counter current in them help in maintaining and increasing osmolarity towards the inner medullary interstitium. The transport of substances facilitated by special arrangement of Henle's loop and vasa recta is called counter current mechanism. So you have to study what is counter current mechanism and what is the how this mechanism works, or how the glomerular filtrate gets concentrated, that is how the urine gets concentrated. Hope you all understood what is the mechanism of urine formation and the mechanism of concentration of filtrate. That's all for today. Thank you.